And I think we should be recording now, which is a good place to be at. So sorry that we started today just a couple of minutes late because everything's running a little slow today with my computer. If problems continue, then we'll just have to reboot. But I'm trying to avoid that to the very, very end that I can. So we just can keep going nicely and smoothly. So this week we'll be focusing on blood vessels, but before we do so, I just wanted to cover a couple of general things as well. Let's see. Yeah, like I said, everything's a little slow today on the computer side of things, but I just want to give an update about the lab practical one and also about the exam one. Last week we did the PhilS labs, which were a little different. Some students were successful on completing those, some weren't, and I have been promising that I have an alternative plan for that as well. So I want to give an update about that as well. In terms of missing retaking announcements, that's assignments, that's something that we've talked before. But really what I want to focus this week is to go through the blood vessels. We'll look at the general structure of a blood vessel, and then we'll talk about the kind of physiological adaptations of blood flow, blood pressure, and pulse in particular. And then I have a little bit of information to share with you about the special circulations and clinical aspects that I'm going to more of share through material that you can study at your own time. But that's the plan for today. And what I wanted to do was to mention where we are with the lab practical one and exam one. So most of the students should have received their lab practical one final grade. If you have not, that's just one of the only very, very few ones that are missing. So I go through all of these 3 times without knowing who's the student, and independently without seeing the previous time scores. So it's very, very consistent from one cycle to another. So lab practical one should be, for the most students, there should be grades up there. Exam one, the last cycle should be finished today or tonight. So I'll make an announcement of that they're ready. But if you don't see comments, if you don't see your grade having changed, I have not covered it yet for the lab practical one, but with most students I have. And overall, I just want to really say thank you for the great work. There were really strong scores. I think you'll be able to see from the corrected exams that I'm very generous with grading as well. So there's a lot of extra credits up for grabs. There's a lot of partial points up for grabs and so on. We'll spend a little bit more time looking through the most common challenging questions or most common hints that I can share with you guys. And what we'll do that next week. So I'm just gonna kind of leave that for now as a bit of a, we'll come back to it. That's challenge to get to the tools because of the screen being so frozen, but we'll save that for the next week. PhilS labs were giving challenge for some, and some they were not. And I've been thinking very carefully how to tackle that. And what we're gonna do, there is gonna be a very, very simple way, and I'll just share it with the entire class all at once through the announcement. So don't worry about that. That's the biggest message at this point. As always, for the missing assignments, anything retaking those, I'm totally happy to facilitate that. You just have to message me directly with the specific details of which assignment are we talking about. I encourage you to kind of try to have a good strategy if you're retaking an assignment at how to do that. And notice that most of the assignments you can still take even after the deadline, and there's no grade deduction. So give it a go as well, that if you're able to run it. I don't lock the grades, So when you go to those, you'll be able to rework on those as well if you feel that that's something that you want to do when you're looking at your overall grades. And this week we have less of a bunch of assignments for you. So we have the familiar LearnSmart, the quiz, and practice anatomy. And all of these are due at the end of the week. And we have also a lab, but this lab is going to be like you saw back in the day when we first started. So familiar, none of this Phil's lab format. So it should run really smoothly on the Macro Hills Connect platform. There should be no harsh surprises there. So I'm expecting that that's going to be easier for most students to access. What I want to really do is jump to the chapter of looking key points, and I'm just not 100% sure was it chapter 20 or 19 in your textbook, but what we will be talking about is going to be the blood vessels. So give me a second as I'll jump around on the screens. Especially with everything running slower, it's just gonna take me a second to get the computer to cooperate with me. I think that really rebooting it would be the way, but I just don't wanna not to finish off our session and have to come back. So bear with me a second. Yeah, I think that it's the chapter 20 in your textbook. In some other textbooks, it's the chapter 19. So that's why I was like, oh, I don't remember which chapter number your textbook uses. But we will be looking at the blood vessels. And this really is the last of all those chapters where we're looking at the cardiovascular system as such. So there's been 3 chapters, and then next week we'll move off to a kind of a complementary system to the cardiovascular system, and especially to the vascular portion. So we'll talk about that as well. And I'm just waiting the things to open up in case you're wondering why the delay. I think I have the grading softwares running on the background, which are turning out to be a little much for the computer to handle on the background. So what I'm gonna do, I'm gonna share them, provided that this is deciding to work with me. Perfect, now we got where we need to be. I'm gonna share them, but I'm gonna share them with you like this. So if you cannot see some of the slides, just let me know. The reason why I'm doing it on this format, not blowing it up for the full size, is that I don't want the little videos that I have at the corner to start running. So you don't have to listen cacophonic twice of my voice. So really, the blood vessels, what we're looking at today, they're going to be the kind of a delivery system of the cardiovascular system that we've had a look before. So we've talked about the blood, we've talked about the heart, and now it's time to focus on these channels or conduits through which the blood will then be transported from one part of the body to another part. So that's our focus for today. And I'm trying to just get the machine to move forward. And there's many different kinds of clinical reasons why we would focus on this system. So we see bleeding, we see bruising, we can see elevated or lower than normal blood pressure. We can see even clots in this vasculature. So there's many reasons why, as a person working to the clinical or moving to the clinical field, you would want to be familiar with this system. And there's really, when I ask from students, there's three types of blood vessels that students always list for me. They list the arteries, which are of course the vessels that transport the blood away from the heart in the systemic circuit. So this blood is rich in oxygen concentration. So it's often illustrated on the diagrams as this bright red system because of the oxygenated hemoglobin having this redder color. Then students talk to me also often about veins, which are the blood vessels that bring the blood back to the heart in the systemic circuit, being rich in oxygen or being poor in oxygen in the blood. So it's a deoxygenated blood that we have in this system. So oxygen has been already used in other parts of the body before it returns back to the heart. And in between those, usually students also know that we have capillaries, which are really the tiniest blood vessels where the exchange from the cardiovascular system to the tissues happens, and then from the tissues back to the cardiovascular system happens. So that's where the big side of exchanging things is. And we end up finding, if we count kind of the amounts of vessel area between all these three, that the capillaries are the most numerous part. So that's the business end, and that's the numerous part. So this is what I normally get from students, but there's going to be two more that you need to know on this level of study. So we're going to add structures known as arterioles, which are basically, if you wish to imagine it, smaller versions of arteries in between the arteries and capillaries. And there are some adaptations that we need to remember with those. And similarly, we have venules, which are smaller versions of veins between the capillaries and veins. And again, they have particular adaptations that we need to know about. So we'll cover all of those 5, but moving onwards from this point onwards, when you are asked what are the blood vessel types that we see in the human, I would hope that you would think of all those 5, because arterioles and venules are just as important as the arteries and veins. We just don't cover them really earlier on the A&P courses or on the biology courses. And what I next want to discuss is a general structure of a blood vessel. So all blood vessels have in general 3 layers that we need to know. And then when we start looking at these more specified types of blood vessels, we'll notice that those 3 general layers of a typical blood vessel, there's slight adaptations from one to another. But to really make the learning process the easiest, we're gonna go first through the general model and then see what's different from one to another. And these layers are known as tunicas. So tunics, and sometimes some students find that really easy to remember. Some students really struggle. I guess that the best I can give as a guy who's not really a specialist of fashion, I think that ladies do wear tunicas sometimes. That's an outer layer clothing that they put on, kind of has an opening for head and arms come out, but really the outer layer or layer of clothing. So as long as you consider that as a term for layer, you'll be totally fine. And these tunicas differ in the last word that they have. So working from the inwards of the blood vessel and moving out, we have first tunica intima, then we have tunica media, and then we have tunica externa. And I think that these are the terms that are most commonly used here in America. In British literature, you sometimes see the tunica externa being referred also as tunica adventitia. So don't get thrown off if you see those two terms being used interchangeably. So I just want you to be aware of that. Sometimes that happens. And when I'm talking about the layers of any structure, really, whether it is a blood vessel or whether it is a digestive tract, really, I often refer to a lumen of a blood vessel. That spells for Lumen. Lumen is basically the central space. So sometimes students just get thrown off because I use that term. Don't let that confuse you. It's just one of those terms that we commonly use on this field. And so let's start by looking from the innermost vessel wall, that what do we need to know about that. And tunica intima, how I try to remember it, or I tell my students the easiest way to remember it is to consider that it is in the most intimate contact with the blood that's passing inside that blood vessel. So that's why it's intima. And it is really made of these simple squamous epithelial cells. So you might remember these from when we reviewed the different types of cells. When it was a simple, it meant that there was one layer. And when it was squamous, it meant, oh, I didn't mean to jump that far. It meant that it was these kind of flattened discs, if you look at the cross section. So really the function of tunica intima is to protect the other layers that are going to be coming afterwards, but also facilitate the passage of materials. That's why it's these flat, thin layers that materials can easily move in and out through that structure or through that layer. And that takes us to the next layer, which is the middle layer of everything, tunica media, kind of in the middle. And that's made of really of the smooth muscle. So there is a muscle layer there, and the muscle layer is going to be important later on when we look at that the muscle allows the blood vessel diameter either to shrink or dilate. So depending on what's the need, and that's a great way to affect then, for example, blood pressure differences, but also of the regulating the blood within the cardiovascular system. In addition to the smooth muscle, there's some amount of elastin, but really what you want to know is that it is the smooth muscle layer which then facilitates this vasoconstriction. And you remember vasoconstriction was when the blood vessel became smaller in diameter, and vasodilation when it extends, opens up larger in diameter. Tunica media is the thickest layer of all, so that gives us a clue that it is a really important layer. And on this level, there's less passage of the materials. And that brings us to the tunica externa, which is gonna be, I'm trying to get there. So that was what I was talking earlier on. You can actually see on that microscopic image all that smooth muscle layer in the blood vessel wall. And what I want to talk now is going to be the tunica externa. Tunica externa is mainly connective tissue. Its function really is to reinforce the walls, anchor them to the site, support the blood vessels. And in addition, we have there, in addition to the connective tissue, various nerves and lymphatic vessels running. And if it is a really large blood vessel, and again, I'm trying to move ahead, sometimes we see this structure called vasa vasorum, which is really these blood vessels that come from the blood vessel and supply the blood vessel wall itself. so nourishes that. So if you think of the coronary arteries that we talked earlier on in a previous chapter, we saw that just like any other tissue, the heart's muscle wall required blood supply. So does a blood vessel. And if it is a really big blood vessel, it becomes harder for that blood vessel to receive the blood supply from inside, from the lumen of that vessel. So instead, we see this adaptation of little vessels supplying the heart, supplying the blood vessel wall itself. And those were known as vasa vasorum. So what I have here is really a good listing of these 3 layers that we should know in terms of the blood vessel structure. And this image might look really busy, but this is actually all the stuff that we have already covered. So it just really nicely lists them and shows that we see them both in arteries and veins. But really, you don't need to take anything else away of this image just yet. What we'll do next, we're going to start comparing the arteries, arterioles, capillaries, venules, and veins between each other. And we'll see how these 3 layers, tunica intima, tunica media, and tunica externa, or sometimes called tunica adventitia, how they differ from one blood vessel type to another. So without further ado, I think we're ready to jump in there. And let's start with the arteries. So what's special about arteries? Arteries are typically characterized by their size, so they're thick, and their function. And size and function, or anatomy and physiology, go hand in hand. Because what really arteries do, they transport that blood that leaves the heart right immediately after blood comes out of the heart. So that blood is going to be in high pressure because the blood gets squashed out of the heart very strong, so that it has that force to do that entire loop that we saw on the systemic circuit. So there's going to be a very high blood pressure. So for that reason, the arteries are very thick as well. The other thing that we see is that the blood pressure or the blood flow within the arteries is also usually slightly irregular. So it's this pulsatile flow. When the heart contracts, and especially the left ventricle contracts, blood shoots out at the high pressure. And then when the left ventricle relaxes, the blood pressure might drop a little. And then it shoots up again, and then it drops and shoots up and drops. So for that reason, arteries are really good at handling this variation, this pulsatile flow. So that adds to the thickness of them and their wall adaptations. One way how you can tell arteries apart is really if you work with catheters, they are on these dead individuals who we have emptied of blood. They feel much thicker than the veins, so you can just physically feel it apart. Sometimes you can see it, but most often what you'll see anatomists do when we do postmortems, we just feel them, different vessels, and you develop that feeling that, oh, this feels much thicker, this feels much more elastic, this probably is an artery, even before we start following where that blood vessel goes or where it comes from. So it really is something that's quite noticeable. And here I have a really, really nice picture showing the difference between arteries and veins. So on the right-hand side of this microscopic image with the white arrows, we'll actually see a vein. And then on the left-hand side, we see an artery. And if you compare their walls, we can see this dark blue band in the artery wall. And this is the elastic layer. And we'll see that the artery has also maintained its shape. Even when these blood vessels have been emptied of blood, we've sectioned from them for the microscopic image, they retain their shape really well. And that's because of this high amount of connective tissue on the walls of arteries. Whereas the vein on the right-hand side, it's kind of started to collapse since it's empty. But what I want to do next, I want to talk about 3 different types of arteries that exist and how arteries can be classified. So the first group is the group that we see usually immediately after the heart. So not only it's the first in our review, but it's first in the order of when we go through different kinds of arteries after the heart. So that's going to be our elastic arteries. And elastic arteries have, let me just see, do I have a better slide? There's a really nice picture of elastic artery. So they have a very, very thick wall. This lumen facilitates, so the center part, lumen facilitates the flow. So there's very little resistance for this flow, but they don't really deal with the passage of the blood through these elastic arteries. So they are often known as conducting arteries. So their function really isn't to allow for this exchange from the blood vessel to the tissues or from the tissues to the blood vessels to happen. They simply conduct the blood from one place to another. And a good example of an elastic artery would be our aorta and its branches. So the first blood vessel that comes out from the heart right after the left ventricle. So there we see a really good example of the elastic artery and how it's functioning mainly just to conduct it. So there's a lot of elastin in the wall of these arteries, and there's really very little vasoconstriction. So their size cannot be brought down all that much. They do expand and they recoil back to the normal. And the function of that is that if you think like an artery, the first blood vessel after the heart and after the left ventricle, there's going to be a lot of this variation in blood pressure, and also the blood flow is going to be pulsatile, as we saw. So by having this elastic wall that can expand and recoil back to its size, that helps us to kind of stabilize some of that pressure, work as a reservoir for these pressure fluctuation, but it never really starts contracting smaller than what it is in size. So that's a really quick overview of the elastic arteries. If you want to kind of describe them somehow, I would really use that term that they're like conducting arteries to guide you. So what we're going to look at next is going to be a second type of arteries. And this second type of arteries comes after these conducting elastic arteries. So we have gotten a little bit further from the heart at this point. So these are going to be our muscular arteries. And again, I have a nice diagram of a cross section of a muscular artery here. And what you'll notice that the pink layer kind of roughly in the middle, so tunica media, is gonna be thicker. And actually most of the arteries that we study and that we name are gonna be these muscular arteries. And these are gonna be distributing arteries. So just in addition to just conduction, they also play an important role in distributing the blood to various different parts of the body. So they're really kind of exits of a highway, if you wish. So they direct the blood where it is needed in different parts of the body. They do have, like I said, very thick tunica media. So the smooth muscle layer, in fact, the thickest of all these types, and there's a lot of this smooth muscle but very little elastic tissue. Again, if you look at this diagrammatic drawing that we have here, the amount of blue is very thin, so there's not that much of elastic tissue present. What they do, because there's a lot of this smooth muscle, they can actually vasoconstrict. So they can become smaller and significantly smaller. if we need to, for example, to manage blood pressure or controlling where the blood flows and so on. And the last ones that we have left are going to be the arterioles. So arterioles were listed in my previous listing as a separate. It is in between the arteries and capillaries. So it's kind of depending on how you wish to classify them. So what we need to know about arterioles, they get smaller and smaller, both in diameter, but also in the thickness of the vessel wall. And eventually it's just going to be a single layer of cells. And this takes us to capillaries and capillary beds. But what we notice that before we reach these capillaries, these arterioles play an important role in controlling which of the capillary networks are open. And when we talk about a large network of interconnected capillaries that move from arteries to the veins or arterioles to venules, we talk about capillary beds sometimes. So it's like a bed or network of capillaries. And what you'll notice or end up noticing if you do some physiological calculations and adding up the cross-sectional area of arteries, arterioles, capillaries, venules, and veins is that the diameter in total of the capillaries, all the capillaries in body, is so large that we actually don't have enough blood to have blood in all capillaries in your body at one time. So we always have some of these capillary networks open and some of them closed. And the opening and closing of this happens by these small muscle bands that you might see on this diagram. I admit it's small, right where an arteriole ends and a capillary starts. So these are like small circular bands of muscle tissue, smooth muscle, that can either close or open a particular capillary network behind it. And that's how our body controls where do we want the blood to go to, since there's not enough blood to go to everywhere. So when you are, for example, physically active, we direct blood rather to the capillary networks that are related with the skeletal muscles, movement, all these functions that are required for large bursts of energy. So a lot of oxygen is picked up from the lungs. So we open those capillary beds. Your heart is very well supplied. We close off networks that are not needed at that time, like digestive system would not be at the time of strenuous exercise as heavily required, or urinary system. So that's the idea, that we direct the blood wherever the blood is needed. And that brings us quite nicely, now that we've seen the last part of the arterioles with the precapillary sphincters. So the proper term for these is that they are sphincters before capillaries, so precapillary sphincters. So now we can move on to look at the arteries a little bit. And what you see on this is one capillary network fully closed off. So in this case, the blood would just simply pass from an arteriole to venule through this one single capillary. Unlike what we saw earlier on, where all of these precapillary sphincters were open and we were directing blood to this area, and we were probably doing a lot of exchange of materials. But now we're simply letting the blood to pass through it and not directing that blood to do exchange. So now we get to the capillaries. And capillaries are, like I said, the smallest of these blood vessels that we see. They're really, really small. They're about a diameter of one single red blood cell. And sometimes they can be even a little smaller than the diameter of a single red blood cell, because red blood cells can squeeze through if they're normal, healthy red blood cells. So it allows the passage of smallest of materials. And this is important because we really want to make them as small, as short of a distance to pass across them, and so on. So what we end up finding on capillaries, that there's really just a single layer. The only layer that we see on those is going to be our tunica intima, so the innermost layer of that general structure of a blood vessel. And their function is really to supply the cells and interstitial fluid, tissues with oxygen and nutrients, and pick up then waste products and carbon dioxide. There is an exception in few places, for example, eyes, cartilages, and so on. But we can kind of summarize that their big function really is the exchange of the materials, the passage of materials. And there's going to be 3 types of capillaries. And this might sound now like, oh my gosh, there's a structure and a substructure, but these make a lot of sense. So I'm trying to convince you not to worry about it too much. So the first type of capillaries that we're going to look at is going to be called continuous capillaries. Continuous capillaries are found, for example, in the skin, muscles, lungs, central nervous system, also blood-brain barrier that separates the general cardiovascular system from the circulatory system of your brain. We don't want toxins to cross there. And what we see, their wall, this tunica intima, made of simple squamous endothelial cells, it's really continuous. There's no gaps. They're packed one next to another. So it really is, like the name describes, a continuous capillary wall. And this one makes sense when we look at the next one, which is gonna be then our fenestrated capillaries. And these are found, for example, for kidneys, intestines, places where hormones are secreted. The term fenestration, if I don't remember wrong, I think it's from French, and fenestration refers to like a little window. So you can see on this wall of this type of capillary that there's little windows in these simple squamous endothelial cells. So these little windows allow for materials to be exchanged even easier than in the continuous capillaries. If you wish to describe it somehow, I kind of think that it looks a little bit like Swiss cheese with all these openings in the wall. So hence the fenestrated, which means the windowed or rich in windows capillary type. And the last one that we're going to look at, you can guess already probably that it's going to go even more extreme. So sinusoidal capillaries, we find them in liver, bone marrow, spleen, adrenal medulla, and places like that. What we have there's these huge gaps of this simple squamous endothelium missing. And the amount of this missing tissue there actually allows a lot of exchange to take place. In addition, by having these huge gaps missing, the wall inside these sinusoidal capillaries is no longer as smooth, so the flow of blood slows down significantly as well, which again allows for more exchange, more processing at these kinds of capillaries. So that's probably a really good summary of the difference between the 3. And as always, feel free to shout out if I go through anything too fast or too slow. That the function of these sessions is really to provide you a chance to ask questions as well. And the last really set of structures in terms of the blood vessel anatomy that we need to look at is going to be the veins. And veins were always the ones that carried the blood to the heart. So what we end up finding if we compared the arteries and veins, you remember that microscopic picture earlier on, they have much thinner wall, and they also have much lower pressure, because you remember the high pressure was immediately after the heart. And now that we've gone through the entire systemic loop, the pressure needs to drop by the time we get to the capillaries. If there's too high of a pressure in capillaries where there's only one single layer of simple squamous endothelium that would burst the capillaries. That's not great. So the pressure drops by the time we get to capillaries. So after capillaries, it's really gonna be a low pressure also in the veins. The pressure can't just suddenly pick up. And in fact, most of the blood vessels in our body are actually veins. So veins make a significant portion of what we end up seeing. So often I ask from my students in face-to-face classes that how does blood get back to the heart in veins? And I get many different kinds of answers, but to consider this, let's consider that we're standing or sitting upright. Our heart is here in the thoracic cavity, but we have transported blood all the way to our toes. That's been easy because there's that high pressure pulsatile flow of the blood out of the heart, and then it travels to the capillaries in your toes. But how does the blood get back from your toes to your heart? If we're standing upright, even in terms of the gravity, there's a huge job to climb up this way. And I often hear many different kinds of interesting theories from my students, for example, that, oh, it is the force of the blood pumped out of the heart that's pushing rest of the blood back to the heart. That's not the case. I often also hear that they propose that the heart sucks the blood in through these veins, and that's not really the case. The secret for getting the blood back to the heart from the veins is really an adaptation that we see on the walls of the veins. And this adaptation is gonna be having valves in there. So just as we had heart valves that separated one chamber from another, we have valves within the veins. And the function of the valve was, of course, to facilitate one-way direction of the blood within that structure. So blood can only go to one direction. If it tries to go back to another direction, that valve is closed and prevents the backwards flow. Well, we end up finding these valves in veins. So whenever the vein, which had a thin wall, gets squashed by structures around it, the blood gets squeezed on both directions, but it cannot go backwards because there's valves stopping it. So the only direction where the blood can go is further along to the direction towards the heart. So that squeezes the blood up a little, and then when this cause of squeezing relaxes, it won't fall back because there's another valve there stopping. So what we find is that the veins are often surrounded or often in locations where there's a lot of skeletal muscles around them. So we constantly move, we constantly squeeze these blood vessels. So the constant moving allows the blood to move a little by little back towards the heart. And the valves make sure that it only moves to that direction. And what we end up finding is that we actually move a lot in our everyday life. Even when we're sleeping, no one sleeps perfectly still. There's constantly a little bit of moving. And this is how we're able to return the blood back to the heart. And this is known as venous return. So veins returning the blood back to the heart. And sometimes to understand a concept, it's easier to think of when it goes wrong, and we'll do that. But I want to just highlight, like I said, there's a lot of veins in many parts of our body, the most common type of blood vessel, and they're often in locations where they get squashed easily, so you can see them. Also, there's going to be veins, for example, in the thoracic cavity, so whenever you breathe, they get squashed. So it doesn't just have to be the skeletal muscles. It can be any other movement that helps the blood to move a little bit. And like I said, we're going to look at a clinical situation where this would not be working. And this would not be working, for example, if a soldier is standing still in a parade for a long period of time, for an extended period of time. So soldiers are required to be very, very still. And if they're not doing that, they're not doing as much of that's this venous return of the blood back to the heart. And as a result of this, they might pass out. So standing with your knees locked, not moving for a long period of time. If you're not getting blood back to the heart, you're not also having that new blood from the heart to pump to other parts of the body, including your brain. Your brain says that, hey, I need more blood, and you're not moving. What's the easiest way to quickly rectify that? Well, let's bring this person in a flat horizontal situation, because then the blood has easy time flowing everywhere. So that's your body preventing a damage to the brain. It's useful that you pass out, because this is how we ensure that this venous return, if it's not happening, we find another way to quickly get the blood to replace whatever blood is missing from the brain. The other thing that you might notice with these is that sometimes we have varicose veins or spider veins. This is when we have in vein valves, but some of these valves fail and blood ends up pooling on top of one functional vein. So now the walls, because they were thin in veins, they end up stretching, becoming wider, and blood pools, and those are what we see as those varicose spider veins. And there's a lot of debate what causes them. There's some hereditary reasons. Some you inherit a likelihood to develop varicose veins. Certain conditions also might play a role. Standing a lot, aviation-wise, being in high altitude, elevated vein pressure in the veins. So there's many functions that contribute to those, but really it is the failure of these valves that then allows the blood to pool. And what I wanted to say next is that we're going to move on and have a look of the blood flow. So more of the physiological aspect of this. And blood flow is measured in an amount per a certain time period. So usually in milliliters per minute. So that's the amount of the flow that we end up finding within a particular part of the cardiovascular system. And I want to move on to the next concept, which is going to be even more important. That's going to be our blood pressure, often abbreviated in medical notes just as a BP. So that measures the amount of force that the blood is pushing against this wall of a blood vessel. And often is measured as millimeters per mercury. And when you think about it, that what's a typical blood pressure of an individual, it varies a little bit. We have been changing this also in the literature when we say that what's a healthy blood pressure, but it's roughly around 120/80. So we see two numbers. Sometimes it's listed as a little higher, as a little lower, but roughly around these numbers. So we see two numbers, the upper number and lower number. Why would we have two numbers for the pressure that the blood exerts against the wall of a blood vessel? Well, that's because you remember the pulsatile flow of the blood. So we have the systolic value, which is when the left ventricle especially is contracted, and then we have the diastolic value when the left ventricle is relaxed. So that's why we get the two pressure values. And if you're wondering what I'm looking and laughing at, it's my cat who's playing busy with the toy mouse. So the next question would be then, I think logically, that what affects the blood pressure? And let's see if we get moving along. So we saw this systolic and diastolic value. So systolic was during the contraction, squeezing of the ventricles, and diastolic when the heart walls, ventricles especially, are relaxed and filling with blood. So there's many factors that affect blood pressure. For example, the total amount of blood in your cardiovascular system. So if you lose a lot of blood all of a sudden, say as a result of an accident, you will have a drop in your blood pressure. Also, blood pressure depends on how much your heart pumps out blood at one go. So the cardiac output, which we calculated as a combination of the single contraction volume of a heart contraction plus the number of contractions in a minute, so heart rate. So cardiac output was that. Also, the resistance within the to the blood flow within the cardiovascular system, especially in the vasculature. So that's known as peripheral resistance plays a role. And of course, as well, how well the blood flows in vessels, how elastic the vessels are, and so on. So really, we can calculate your blood pressure as a sum of multiplying the cardiac output with the peripheral resistance, this resistance to the flow of the blood in the vasculature. And some people say that, oh, I've got pretty good blood pressure, but when I go to see a doctor, my blood pressure spikes up. This is sometimes referred as a white coat syndrome. So when you're in a stressful situation or a nervous, whether you're seeing a doctor or whether you're seeing a nurse, your blood pressure might go a little up because you're just nervous about that. So normally what we want to do when we measure the blood pressure in clinical practice, we want to have the patient seated for a while in the seat. We actually want to take the blood pressure from the level of the heart. That's why we typically put the cuff to the upper arm. Ideally, we would measure that 3 times and then take the average of those 3. If you have white coat syndrome and your blood pressure is elevated when you go and see a doctor or a nurse, we might give you a blood pressure cuff or ask you to get one at home, and then you measure it in your comfortable own environment. And what we end up noticing is that a lot of the time it's gonna be lower. Sorry about that. I had a notification pushing on that wouldn't stop. So next thing I want to talk about is going to be pulse. So we can measure pulse from the arteries. So the arterial blood pressure, we're looking at that fluctuation between systole and diastole. How many times we see that fluctuation in a minute? So pulse is really a result of when the ventricles push blood to the aorta and in the systemic circuit, and the blood pressure shoots up, and then when ventricles relax and blood pressure drops. So that's really, if we think of the layman's terms, our heart rate. We can get a clue of your heart rate without having to cut in by simply measuring this pulse. And typically it's somewhere around between 60 to 100 beats per minute. It varies from an individual to another. It varies on the level of your exercise at the moment. It varies depending on your health level and so on. But that's the typical range. And there's many places where we can actually measure the pulse from. So these are known as our pulse points. So the most common one probably is from the radial artery at the wrist, but we have a bunch of others that we could also look. And full list of those is in your actual lecture video. Then I wanted to talk really quickly before we wrap up about the regulation of blood pressure. And there's both short-term and long-term regulatory mechanism. And this is where, having learned that homeostatic control mechanism loop becomes very useful. I said that I'm going to explain a lot of things with that. So let's have a look. So the stimulus, if we have a change in blood pressure, the stimulus would be, of course, our blood pressure. And the typical blood pressure was somewhere around 120 over 80. So that's going to be our set point within the brain, within the control center. Well, how do we change any notice any changes in the blood pressure? Well, that's going to be through a system called barochemoreceptor reflex, sometimes more just simply just called baroreceptor reflex. And what these are various receptors at different parts of your body. For example, at the carotid sinuses, on those dilated areas, for example, where the common carotid vessel divides in the neck, in aortic arch. So on the arch of aorta, we have also receptors there, and in various large vessels. Well, what happens then? We notice that there's a change. The message gets passed to the control center, which was our brain, and within the brain, particularly the medulla. So medulla oblongata, that structure, that blob in the brainstem. And what we do, we can act. An effector can be through various different ways. It can be through medication. It can be to treat the cause. We can also do vasodilation or constriction depending on whether it's too high or too low. So you see how the homeostatic mechanism loop becomes useful in explaining complex concepts in biology. And if you feel that this went too fast, again, I summarized this in full or explained this more in full in our full lecture videos that you can find on the lecture materials. So really, what is the structure that would control the blood pressure, and not in this short-term manner that we just saw, which was like an immediate response? Let's take medication that acts immediately. Let's treat the cause that acts immediately. Let's do vasodilation or vasoconstriction. But if it's a longer-term issue, how do we regulate the blood pressure? Well, that's going to be through kidneys. And I highlighted that was really important. The kidneys, their main function is the regulation of blood pressure. So what we can do if you have too high blood pressure, well, let's get rid of some of the fluid in your body, especially fluid in your cardiovascular system. So let's produce urine and get rid of that urine. So that would bring down the blood pressure because there's less liquid now. The other option would be if you have too low blood pressure. So in that case, we would want to conserve water so we don't get rid of any water. We maintain the little water that we have in the blood. So that way we're able to preserve the blood pressure if it's too low. And there's a very complex mechanism for the long-term regulation of the blood pressure. But all I want you to know from those diagrams is that we have a direct mechanism and indirect mechanism. The indirect mechanism is much more complicated, and that's sometimes referred as renin-angiotensin-aldosterone mechanism. You don't need to know it in detail, but that's just linking up some of those hormones that we saw before when we were talking about endocrine system. So really what we want to have high enough blood pressure, but not too high. That's if you want to summarize what really happens here and what matters. And as the last thing, I just want to mention that, of course, our blood pressure is affected by various different factors. Your age, sex, weight, even your race can play a role. Our posture, how well you sit. For example, where we're measuring that blood pressure, so that would make an effect if you're sitting with a poor posture or good posture, your level of physical exercise at that moment, or how healthy, how fit you are, your emotions. If you get upset, your blood pressure can spike up, your mood, your medical state, if you have high fever, for example. So blood pressure is affected by many things, but it also tells about a lot of things. So that's why we try to measure the blood pressure. It's a great initial indication about what's going on. And sometimes we have some issues with blood pressure, and we're going to look at two cases, too high and too low blood pressure. So hypertension is when you have too high blood pressure. And I think the latest I saw in the clinical use, it was classified as 140 over 90 millimeters of mercury. But these values, like I said, change a little bit depending on what's the recommendation, how these are set. Why is it dangerous to have a high blood pressure? Well, the risks become because you can end up with heart failure, you can end up with vascular diseases, renal failure. So when the kidney filters blood, there's too much of a blood pressure that can damage the kidney, also stroke. So in these conditions, when you have hypertension, your heart ends up having to work much harder, your myocardium becomes larger, but even though it grows in size, it's very weak and it's very flappy, and plaques can end up building up. Some of the risk factors that can make you prone to have hypertension, so high blood pressure, it is hereditary, so you inherit that, but also diet that's rich in high in fat, high in cholesterol could be an issue. Obesity. So one way to bring down your blood pressure, if you don't want to go on blood pressure medication, would be to try to drop some weight. Your age, having diabetes, being very stressed, smoking as well. So there's a lot of things that we can do before we have to rely on medication. Often there's no clear cause. It can be a summation of these or just we never know. But really, there's a significant role that lifestyle plays. So you can really affect that quite a lot, quite easily, quite effectively. The other extreme is when we have too low blood pressure. That's known as hypotension. So that's often classified somewhere below 90 over 60 millimeters of mercury. Usually this is not that much of an issue unless there's inadequate blood supply to tissues. Some even have speculated that this could be beneficial and associated with long life of an individual or not having cardiovascular illnesses. There's subgroups of hypotension. It can be just temporary, dizzy when standing up, for example. It can be chronic, so often due to poor nutrition, and that comes with risk of developing other conditions, of course, if you're not getting good nutrition. Or it can be acute and indicating shock, that you're about to go to shock. And I do want you to know the signs and symptoms of shock. And you'll notice that British people love abbreviations, mnemonics, in terms of that. Police constable is what we say rather than cops in UK. So it's going to be, our mnemonic is going to be PC FATS. Police, that's fat, eating donuts. So the signs and symptoms of a shock is that person's skin is gonna be pale, it's gonna be cold and clammy, their breathing is gonna be fast but very shallow, same as their pulse is gonna be fast but weak. They might appear agitated, they often are thirsty, and that means that they're going to shock. So PC FATS, that's the signs and symptoms of shock. And I usually like asking about that. I would ask you also, if we were in a classroom, what would you do with someone who's going to the shock? The big thing is that we don't want them to collapse, hit their head, so we might lay them down, raise their feet, so we direct more blood to the head, loosen clothing, any constricting clothing. We want to calm them, monitor them. But of course, it comes down to what is the cause of the shock. Let's treat that. And if you have a large blood loss, for example, you do tend to go to shock. And shock really is a medical condition, not how we see those being used as shock sale and stuff. No one goes to a shock because they're seeing a sale. How do we control the blood flow and make sure that it's constantly controlled? There can be separate branches. You can have nervous system and hormones playing role. That would be your extrinsic control, but also meeting the local needs in a kind of intrinsic level. It's really this capillary control that we saw earlier. And let's look at the flow just really quickly. Where most of the blood flows in a normal situation, about 20% of the blood is directed to the skeletal muscles. But then when you're exercising, it can jump up to 70% of the blood being directed to the skeletal muscles. So we talk about autoregulation where we have the local control. Blood flow within to the brain, there needs to be a constant flow there. So it can't handle too high or too low blood pressure because the neurons die very easily. They get damaged very easily. So that's something to keep in mind about the blood flow to the brain. Blood flow to the skin, well, we direct blood to the skin often to regulate your temperature. So for thermoreceptor, thermoregulation. So if we need to get rid of heat, we direct a lot of warm blood to the surface of the skin, and it gets lost from there. You might sweat, so sweat evaporates, catches some of that heat, and so on. If you are losing too much temperature, we eliminate the amount of blood flowing to the level of the skin. Also, skin works as a great reservoir for a lot of blood. In terms of the lungs, we want to have a low blood pressure there. So lungs and the blood vasculature there cannot handle high blood pressure, otherwise it will burst those thin walls where the exchange happens, and we will have blood building up to the lungs. That would not be great. And we already talked about the blood supply to the heart, how it needed a constant supply. So really the last thing to kind of mention is the speed of flow. Where do we see blood flowing fast, where it flows slow? The fastest flow is at the aorta and slowest flow at the capillaries. So if you think aorta, that's where the blood gets pushed out, so that's going to be fast flow. And then capillaries is where the exchange happens. So we want the speed to be very, very slow. So it kind of picks up again when we go back to the veins. This is an interesting interesting graph. We've mapped on top of those structures the total cross-sectional area of the vessels. So you'll notice that the largest cross-sectional area of the vessels is, of course, at the capillaries. That's where we have the most area in the cardiovascular system or vascular system. That's all about exchanging of these materials. So really, if we look at the capillaries, that was the site where we keep these walls very thin. You see a red blood cell there within a very small, thin capillary. We already talked about these precapillary sphincters. Just to remind you, they play an important role in the control of what capillary networks are open, what are closed. In terms of the exchange of the capillary, what happens in capillaries, how the gases, nutrients, everything is exchanged, it's through the diffusion. So the passive movement down the concentration gradient from high area to a low area. Some go through fenestrations, and also there's some amount of active transport. So through the pumping, through various vesicles for larger molecules. In total, we have the same amount of blood leaving and returning to the blood vessel in an ideal situation. The force that pushes blood out of the blood vessel is going to be our hydrostatic force. And the force that pushes blood that would otherwise leave the blood vessel back to the vessel is going to be our osmotic force. So those should be in balance, and the blood stays nicely within the blood vessel. How is this relevant? When we have more fluid leaving the blood vessel than returning back, we end up building an edema. So that's the interstitial fluid components of the blood escaping the blood vessels because our hydrostatic and osmotic pressures are not balanced. And just a couple of points that I really want to highlight as a kind of a key points to remember from this system is that arteries run typically deep, so they're well protected, so we don't end up cutting them. And veins are more superficial, there's low pressure, remember, we kind of want to get them squeezed so we can pump the blood, but we don't want them to be deep in the body where they would be squeezed all the time. The other thing to remember that veins, there's many alternative routes with veins. So those are known, the fancy word for that is anastomosis. So we have multiple ways to take the blood back. And then with the brain, that remember there was a unique drainage system for the blood from the brains. And with intestines, remember that it is the liver where most of the blood goes immediately from the digestive system, and where blood kind of gets filtered. That's because that's where we store the energy. So only thing that we have not covered now is really, other than just quickly mentioning these special circulations that we went through, we have not covered in great detail actually drawing these blood vessels. And for that, let me just jump to another view. For that, I've actually uploaded really nice diagrams on your Canvas weekly module. And I'm leaving those diagrams for you to have a look by yourself, because there's really no smart way other than memorization of the major blood vessels. What helps you is that, remember, you can have all the notes that you wish in your exams, in your practicals. So don't let that freak you out. Don't let that worry you. Use the materials that you have. In terms of the diagrams that I provided, I think I've tried to be very generous. Ooh, now I just need to get the commands up, so bear with me. I've been very generous with different kinds of diagrams that I provided. My goal is to make sure that you find a diagram that works for you. I'm just trying to get to the point where I'm sharing my screen with you again. So when you go to the weekly learning module that I prepared for this week, you will find a bunch of blood vessel diagrams. And I encourage you to make a copy of those, or save those, or print those, whatever you feel is the best way for you to have those available in case we would be talking about them later on, or you would need them later on. You'll see that I have these very schematic diagrams where from here, from the heart, blood would be leaving. We have our coronary arteries that we talked earlier, and then the arch of aorta, from where blood goes to various different parts of the upper limbs and head, but then also going down thoracic and abdominal aorta to all other parts of the body. If you're not a diagrammatic person, I understand that. You're not that engineer oriented. This is the same diagram laid out on top of a drawing of a human body. And again, these blood vessels are named at the most important ones on that. Again, a diagrammatic representation of the blood flow from the arch of aorta and going especially to the head and neck, and especially to the brain. And then you can see it also superimposed on a diagram of a human body. Same for the brain, supply of the blood within the brain. This is the upper limb diagrammatic representation. Heart would be here, the arch of aorta, and we're looking just on one side, the major blood vessels that you need to know of the upper limb. Again, if you prefer more of a superimposed diagram that's more of an anatomically accurate, you have it available. Same for your abdomen. Both the diagrammatic and very anatomically accurate food versions of the different levels of the depth. And then we have the lower limb. Again, this kind of a diagrammatic representation, this would be the abdominal aorta ending and being divided into two common iliac arteries that then continue all the way to the toes with the different names of these vessels. So again, the diagrammatic and superimposed. We also have the veins, so return of the blood, same for those. I emphasize the importance of arteries because arteries are the main arteries always very consistently following. We find them same in the human body in every part. With veins, there's a little bit more variation. They're not exactly always where you expect them to be. So if you're working with catheters, with dead bodies, you'll end up or with patients, you end up sometimes finding a little bit more variation in veins. It's not always quite the textbook accuracy. So that's a great resource that I really wanted to share with you. You'll find it on your weekly learning module on Canvas. So under this week, I strongly recommend that you make that available for you if you feel that that could be something that's helpful, if you feel that you might end up needing it later for the study material, completing those assignments, for the tests, for the anatomy practical, for things like that. And the very last thing that we have left to do after this review is just to have a really finish of the weekly kind of a PowerPoint that I prepared, and I'm just opening that in case you're wondering why is it taking a while. That's, like I said, my computer is running on a little bit of the steams. It's not quite as fast as I'd like it to be, but we'll get there. Let me share that and we can wrap up this. So we have just covered the blood vessels chapter, the key points on there. And I just want to remind you of the tutoring being available through the Cranium Cafe, so the virtual format. Until we're ready to return back to the campus, you can pick the tutor you like. I'm also available if you have any questions. I've had actually quite a few one-on-one sessions in the past few weeks with various students. So if you have any questions, whether they're technical or content related, I'm so happy to schedule. I'm usually available pretty much whenever you just give me a shout out. I'll make myself available for you, even with a short notice. And finally, if you have any questions, I'm happy to answer, always in these sessions, but also always through messages, always making one-on-one time if you don't want us to ask them in public. And that really covers everything that I wanted to address during this weekly session. I just want to remind you that I will be posting an announcement really soon about the lab practical and exam one, that the grades are ready. I will be also posting an announcement about what to do with those fields labs from last week. So don't stress if they didn't work out for you. For most students, they did. But if they didn't work out for you, there is a plan in place. So that's all I have for this week. I'm going to pause the recording now. If you have any questions, don't hesitate to message me. And if you have any questions, Sarah, just feel free to ask. But that's all I have for now. And now that we have covered any questions that we had on a one-on-one basis, I'm going to stop this recording from this weekly session. I really appreciate that you joined me. And as always, remember that there's 2 sessions that I typically offer on Wednesdays, both in the morning and evening. But if you miss those, there's these recordings available. And still, if you would like to have one, you can always contact me and I will definitely find the time to schedule one with you. So let's make it a great week. You're doing great progress, and I'm really excited to be able to share the grades from the exam and practical one with you real soon. So that's it from me for this week. Make it a good week. Thank you. Bye.